This is why you need to know about bacteriophages. They're bacteria's worst nightmare. Imagine a world where bacteria, those tiny organisms that can cause so much trouble, have their own predators. These predators are not other bacteria or larger organisms, but viruses. Yes, you heard that right. Viruses that specifically target and destroy bacteria. These are called bacteriophages, or simply phages. They are like the ultimate nemesis for bacteria, lurking in the shadows, ready to strike. Picture this. Viruses that hunt bacteria. It sounds like sci-fi, right? But it's not science fiction, it's a fascinating reality. Bacteriophages are incredibly specialized viruses that have evolved to infect and kill bacteria. They are found everywhere bacteria exist, from the deepest oceans to the soil in your backyard and even inside your body. These tiny warriors are constantly on the lookout for their bacterial prey, and when they find it, they launch a precise and deadly attack. But it's real, they're called bacteriophages, or phages for short. These phages are incredibly diverse, with each type targeting a specific kind of bacteria. There are estimated to be more bacteriophages on Earth than any other organism, numbering in the quintillions. They play a crucial role in regulating bacterial populations and maintaining the balance of ecosystems. Without them, bacteria could potentially grow out of control, leading to devastating consequences for all forms of life. The discovery of bacteriophages dates back to the early 20th century. In 1915, British bacteriologist Frederick Dot first observed these mysterious entities, but it was French-Canadian microbiologist Félix de Herelle who, in 1917, provided a detailed description and coined the term bacteriophage, meaning bacteria eater. De Herelle's pioneering work laid the foundation for phage research and their potential applications in medicine. Phages are like the ninjas of the microscopic world. They move with stealth and precision, seeking out their bacterial targets with incredible accuracy. Once a phage finds a suitable bacterium, it attaches itself to the bacterial cell wall using specialized structures that recognize and bind to specific molecules on the surface of the bacterium. This attachment is the first step in a highly coordinated attack that will ultimately lead to the destruction of the bacterium. They stealthily attach to a bacterium, inject their DNA, and hijack the bacteria's machinery to make more phages. This process is known as the lytic cycle. After attaching to the bacterium, the phage injects its genetic material into the bacterial cell. The phage DNA then takes over the bacterium's cellular machinery, redirecting it to produce new phage particles instead of the bacterium's own components. It's like a hostile takeover, with the phage turning the bacterium into a factory for producing more phages. Then, kaboom, the bacteria bursts, releasing new phages to continue the battle. This final stage of the lytic cycle is called lysis, where the bacterial cell wall is broken down, causing the bacterium to burst open and release the newly formed phages. These new phages are then free to infect other nearby bacteria, continuing the cycle of infection and destruction. It's a relentless and efficient process that ensures the phages can keep bacterial populations in check. Structurally, bacteriophages are fascinating. They typically consist of a head, which contains the genetic material, and a tail, which is used to attach to and penetrate the bacterial cell wall. The head is often icosahedral, a geometric shape with 20 faces, while the tail can be long and flexible or short and stubby, depending on the type of phage. This unique structure allows them to efficiently infect their bacterial hosts. Think of them as nature's own antibacterial army. They are constantly patrolling the environment, ready to spring into action whenever they encounter harmful bacteria. This natural form of bacterial control has been occurring for billions of years, long before humans ever discovered antibiotics. In fact, phages are considered one of the oldest and most effective forms of antibacterial defense on the planet. They don't mess with human cells, just the bad bacteria making us sick. This specificity is one of the most remarkable features of bacteriophages. Unlike broad-spectrum antibiotics that can kill both harmful and beneficial bacteria, phages are highly selective, targeting only the specific bacteria they are designed to infect. This means they can be used to treat bacterial infections without disrupting the balance of the microbiome, the community of microorganisms that live in and on our bodies. Scientists are exploring phages to fight antibiotic-resistant bacteria. With the rise of antibiotic-resistant superbugs, there is an urgent need for alternative treatments. Phage therapy, the use of bacteriophages to treat bacterial infections, is gaining renewed interest as a potential solution. Researchers are studying how phages can be used to target and destroy bacteria that have become resistant to conventional antibiotics, offering hope for new and effective treatments. Imagine a future where superbugs are no match for these tiny warriors. In this future, phages could be used in combination with antibiotics or as standalone treatments to combat bacterial infections. They could be engineered to target specific bacteria with even greater precision, making them a powerful tool in the fight against antibiotic resistance. This could revolutionize the way we treat bacterial infections and save countless lives. Recent advancements in phage research are promising. Scientists are now able to sequence the genomes of bacteriophages, allowing for the identification of specific genes responsible for their antibacterial activity. This genetic information can be used to engineer phages with enhanced capabilities such as increased specificity and the ability to bypass bacterial defense mechanisms. Additionally, phages are being explored for their potential in biotechnology, agriculture, and even environmental conservation. So next time you wash your hands, remember there's a whole microscopic war going on, and bacteriophages are the unsung heroes. Pretty cool, huh? Stay curious, science enthusiasts. These tiny viruses are working tirelessly behind the scenes to keep harmful bacteria at bay. Their incredible abilities and potential applications make them a fascinating subject of study and a beacon of hope in the ongoing battle against bacterial infections. So keep exploring, keep learning, and never stop being amazed by the wonders of the microscopic world. Bacteriophages are not just limited to medical applications, they are also being studied for their potential in agriculture, where they can be used to control bacterial diseases in crops and livestock. This could lead to more sustainable farming practices and reduce the reliance on chemical pesticides and antibiotics. In environmental conservation, phages are being explored as a tool to manage bacterial populations in natural ecosystems, helping to maintain biodiversity and ecosystem health. In agriculture, phages can be applied to crops to protect them from bacterial infections, reducing the need for chemical treatments. This not only helps in producing healthier crops, but also minimizes the environmental impact of farming. Phages can also be used to treat bacterial infections in livestock, promoting animal health and reducing the spread of disease. This approach is gaining traction as a sustainable and eco-friendly alternative to traditional methods. In environmental conservation, phages offer a natural solution to managing bacterial populations in various ecosystems. By targeting specific harmful bacteria, phages can help restore balance in environments that have been disrupted by bacterial overgrowth. This can be particularly useful in aquatic ecosystems, where bacterial blooms can have devastating effects on water quality and marine life. Phages provide a targeted and effective means of controlling these outbreaks without harming other organisms. The potential of phages extends beyond just fighting bacteria. Researchers are also investigating their use in biotechnology, where phages can be engineered to deliver genetic material to specific cells. This has exciting implications for gene therapy and the development of new biotechnological tools. By harnessing the natural abilities of phages, scientists are opening up new avenues for innovation and discovery in various fields. In gene therapy, phages can be used to deliver therapeutic genes to target cells, offering a precise and efficient method of treatment. This could revolutionize the way we approach genetic disorders and other diseases, providing new hope for patients. The versatility of phages makes them a valuable tool in the rapidly evolving field of biotechnology, where they can be adapted for a wide range of applications. As we continue to explore the potential of bacteriophages, it's clear that these tiny viruses hold immense promise. From medicine to agriculture to environmental conservation, phages offer innovative solutions to some of the most pressing challenges we face today. Their ability to specifically target and destroy harmful bacteria while leaving beneficial organisms unharmed makes them a powerful and versatile tool in our arsenal. The future of phage research is bright, with ongoing studies and advancements paving the way for new applications and discoveries. As we deepen our understanding of these remarkable viruses, we unlock new possibilities for improving human health, protecting our environment, and advancing scientific knowledge. The journey of phage research is just beginning, and the potential impact on our world is truly exciting. So, the next time you think about viruses, remember that not all of them are bad. Bacteriophages are the unsung heroes of the microscopic world, tirelessly working to keep harmful bacteria in check. Their incredible abilities and potential applications make them a fascinating subject of study and a beacon of hope in the ongoing battle against bacterial infections. Stay curious, keep exploring, and never stop being amazed by the wonders of the microscopic world.